Thank you very much for coming. I hope you enjoyed your lunch. I'm Yuyao. I'm a PhD student at EMBO EBI with Irene Papatheodorou. And today I will talk about an application of the gene ontology, which is pretty different from other talks in this track. But it is a method we create called ScGOclust, and it is for finding functionally analogous cell types between distant species. So speaking about distant species, I like to focus on example between flies and vertebrates, and particularly between fly and mouse. So these two animals are about 600 million years apart, while as a reference, a human and mouse are about 90 million years apart. So they're extremely distantly related. And if you ask the question of what are the similarities between flies and vertebrates, different biologists will give you very different answers. So for example, a zoologist might look at the morphology, a geneticist might enjoy researching the gene regulatory networks, and then embryologists will compare how a zygote becomes an embryo. However, from our perspective, we mainly work on single-cell atlases, so we're interested in leveraging these single-cell atlases to systematically compare the cell types across remote species. In terms of computational method, currently there are two main computational approaches. That would be an independent analysis and an integrated analysis. So in an independent analysis, you would analyze the data per species before comparing them, and then simply calculate an expression correlation of some selected genes between each pair of cell types. However, in an integrated analysis, it's possible to perform single-cell RNA-seq data integration and calculate the shared and unique clusters between them. So previously, we mostly worked on data integration. However, we quickly found out that both of these methods have big challenges for comparing remote species. So in terms of independent analysis, when a species distance is very large, it's only possible to include very, very few genes, thereby biasing your analysis. And for data integration, it also simply does not work on remote species, such as those across phyla. Therefore, just to summarize this problem, a gene-level feature are insufficient for comparing evolutionary remote species using single-cell RNA-seq data. This is because at such a long evolutionary distance, the genome has undergone complicated changes with genes getting duplicated, getting deleted, or gain of a completely new gene. Therefore, this creates a very incomplete mapping between the features in these two single-cell count matrices that you are trying to compare. Therefore, you basically don't have any anchor to compare the entities. And this fact leads to the phenomenon that any kind of gene-based integration, correlation, or even just comparing a functional enrichment of cell type marker genes at such a evolutionary distance simply doesn't work. So this drives us to ask the question, can we find another feature that are shared and comparable between such remote species? And when we think back of this question, even though mechanistically speaking, the organs and cells from different species have highly diverse mechanisms, however, as a corresponding organ, you would always need to perform a set of similar functions. So for example, if it's a heart, you would always need cells to contract and pump blood, to transmit neuronal signatures, to support the tissue structure, and to perform immunity. Therefore, we put our eyes on the gene ontology knowledge base, specifically because there we have species-neutral annotation in terms of function of genes. And particularly the biological processes terms is highly relevant in this case because it then provides a bridge for a functional comparison between the remote species. So that is the key message I would like to highlight in today's talk, which is using another feature that's related from the topic that you would like to study actually really makes a big difference. So this method, ScGOclust, is a very simple method. So first we would obtain a mapping matrix between the gene ontology biological process terms and the gene. So it's just a simple one-hot encoded matrix. And then we would multiply this matrix with the single-cell count matrix to generate a profile of the cells, but using gene ontology biological process terms as features. And this matrix can be analyzed just like any other feature matrix. You could perform normalization, z-score scaling, dimensionality reduction, and visualization. In terms of a cross-species comparison, we mainly focus on two questions. The first question is which cells are similar between the species. So for this step, we take the scaled matrices from the two species and do a correlation study between them. Therefore, we can highlight the pair of cell types that are showing an increased degree of similarity. And after we identify these pairs, we are able to see which Go terms are actually driving this observed similarity. So the way we do this is by taking the normalized matrix and calculate the differential. It's like analogous to differential expression, but a differential biological process activity. And then after we collect these Go terms and use a alignment method to find their last common ancestors so we don't miss any terms, we are able to generate the final co-upregulated and co-downregulated Go BP terms across the two species in this particular pair of cell type. So to use the Go resource efficiently, we also paid some attention on how to select which Go annotations that we would include in this analysis. So by talking with the curators, we compiled two sets. The first set is the one indicated with the green color. It is the standard set, which means that we only include every term that's been checked by human. And the second set is the more stringent set, which includes a handful of selected experimental, author, or curator evidence, and we exclude things that are not well supported. And here it's important to highlight that it's not the stringent set is better than the standard set. It's just a different perspective if you would only like to include experiment evidence in the analysis. However, these can be highly biased according to the historical focus and so on. So we start to apply this method to many datasets. This is in collaboration with a group working on fly cell atlas. And we first applied it to a fly and mouse heart dataset. So here you see a UMAP visualization of the single cells using Go BP as features. And it's clear that the distinctions between them, the heterogeneity represented in this dataset, is well represented using the Go BP features. Then after the correlation analysis, we will first be able to identify the similarities between homologous cell types, which should be expected, like a positive control. So here, particularly, we highlight the shared terms between cardiac muscle cells in mouse and cardiomyocytes in fly, and also the Schwann cells in mouse and the peripheral neurons in fly. However, interestingly, in the same analysis, we were also to find some functional convergence between non-homologous cell types, which is highly interesting because when you look at what kind of co-upregulated Go BP terms that they share, it actually makes a lot of sense. So the first pair we found is between a mouse cardiac muscle cell and a fly fat body. So what terms are shared there is basically lots of terms related to glucose and fat metabolism. But it makes a lot of sense because both of these tissues have an active role in these kind of energy and metabolism. Therefore, these processes are, of course, highly active in these cell types. And the second pair we found is the pericyte cells in mouse with the peripheral neurons in Drosophila. And it's interesting because here the highlighted terms are related to nitric oxide cGMP signaling. And it suggests that both of these cells, they contract in response to oxygen levels from this signaling pathway. And these are all well represented in literature. So we proceed to apply this to a more complicated data set, which is the fly gut and the mouse stomach and intestine. And here I will highlight the result that we found a subgroup of crop cells in fly, which were not separately annotated in a single cell atlas, but they show a very high similarity with some of the mouse epithelial cell types. So in order to understand what is going on in this subgroup of crop cells, we plotted the upregulated Go BP terms and genes in this subtype compared with other crop cells and compared with any other cells. So it was very clear that in this subtype, terms and genes related to protein synthesis and lipid synthesis are highly active. And what is worth noting is that here we also find the increased expression of a gene called the 2 gene, which is related to the control of circadian rhythm in fly. So we hypothesize that this subgroup of cells, they are regulated by circadian rhythm, and then they have an active synthesis of protein and lipid, which are most likely to be digestion enzymes and signaling molecules and hormones in order to regulate the digestion of food in fly. So finally, for the fly and mouse comparisons, we applied it to a renal system in fly and a kidney dataset in mouse. And here we find some analogies between the principal cells in fly and the different segments of kidney tubules in mouse. So to illustrate this analogy, we were able to find that in fly, the principal cells at the main segment are corresponding functionally to the distal convoluted tubule and connecting tubule in mouse. These two parts mainly have the function of electrolyte homeostasis, which means that they regulate the mobility of iron and water in the kidney of both of these species. But on the other hand, principal cells in the lower segment in the fly Malpighian tubule map to the proximal tubules in the mouse kidney. And the shared function between these two segments are reabsorption of nutrition macromolecules. It's interesting when you look at this mapping, because actually in terms of the direction from inside the body to urine, they are in a reverse direction. For fly, the hemolymph, which is the fly blood, are at this end of the Malpighian tubule, and then after filtration, they would generate urine at this end. So essentially, what happens first is the control of electrolyte homeostasis with waters and ions, and then the reabsorption of possible nutrition to be used by the body. However, for fly, the blood is here. So basically, what first happens is the reabsorption of nutrition molecules, and then later on, in the more distal kidney tubules, it would regulate more of the electrolyte homeostasis processes. So this is very interesting, and this was just impossible with any other method. So finally, to test until where this method could still be useful, we applied it to an even more distant species pair, which is between Hydra and C. elegans. And these two groups, they are 700 million years apart, so they've diverged for such a long time. But applying this method, we still obtained a lot of signals. And particularly, we were able to identify the shared correspondence mostly between neuronal cells, secretory cells, and germline cells. And these mappings are in line with our knowledge about what kind of function is the most conserved across a long evolutionary time. So those are the stem cell-like versions, the neuronal versions. And here the secretory functions were more like a bonus, but we were glad that this method still gave a lot of insights at such a long evolutionary distance. So in summary, ScGOclust is a method that leverages gene ontology to characterize cell type functional similarities, and it is particularly useful for comparing highly evolutionary remote species where all of our current other computational algorithms simply fails to do so. And this method, applying this method, we generated many computational assumptions of the correspondence of cell types and subtypes in terms of function, and we observed the conservation and convergence through these kind of cross-species mapping. And finally, we would envision that using these kind of method with a gene-level analysis synergetically would further our understanding of the similarities of cell types from both of a functional and a molecular perspective. So with this, I'd like to acknowledge my supervisor and our group, our collaborators, all the founding bodies, and in particular, these publicly available data and resources, such as the ScGOclust, FlyBase, and gene ontology, because the whole work is based on these resources. So thank you very much. Happy to take questions.
Thank you. We have time for some questions.
You may have said this and I missed it. So how do you select which genes you include in your matrix of genes versus Go terms?
Yes, so in terms of the genes, so in order to do this correlation analysis, these two data sets need to have the same group of Go terms that we include. So the genes would then be those that have the annotation of these shared Go terms between the two species.
But you're only including genes that are expressed in each of the cell types that you're trying to match?
No, it's everything. So the whole, across all of the single cells
See.
that basically have the Go annotation.
Thanks.
Yeah.
Thank you. Very interesting work. When you compare fly and mouse, they both have very good Go annotations. But when you compare to Hydra,
Yes.
then the Go annotations might be slightly less deep. So how much does this bias your method? Are you biased and then towards the kind of functions which are easy to annotate in such a very, not only non-model, but very distant from the models? So how does it affect the results?
Yes, it's a very good point. So in order to compare the Hydra and C. elegans, we didn't take the Go annotations from like a database like Ensemble, because I remember it was just insufficient. So we actually inferred them using a method called eggnog, which is basically inferred from orthology relationships across all of the tree of life, basically. And I would say also from the analysis of the evidence code of Go terms for different species is really depend on what the community has been working on. So some of them have a lot of experimental data, but some of them mostly have it annotated from gene orthology. So this is indeed, the analysis would depend on this kind of input data that's available, but that's as far as we can go. But here, at least for this example, we are able to show that the method still works, even if the Go BP terms are inferred, and they're not necessarily with experiment evidence or curated. Thank you.
Hi, beautiful work. So my question is, could we use this method to leverage information we have on well-annotated species genomes, for example, the model organisms, to understand more of the function of the less annotated genomes, so the non-model species? And if used for that purpose, what would be the limitations?
Thank you. It's a very good point. So yes, I would say so. However, it would help us to, at least from a functional perspective, compare a very well-studied species with a less well-studied species, given that there would be some Go inference conducted on this less studied species, and there would be a single cell data available and mapped to the genome and clustered at least to an extent that you could identify different cell populations. So it is definitely a challenging topic. But I would argue that this topic is in general challenging for the field of cross-species comparison of single-cell data, because if you just compare them based on a gene level, it would require the same thing. And yes, we're still in this process, and I think the main value of this work is to observe this question from a different perspective, which is the function perspective. While we already have these available information from these amazing ontologies that basically collects this information for us, then it generates new insights that any other method simply cannot do. Thank you.
So we have time for maybe one more question. If somebody has a question. There's no more question, then thank you very much.